Dear students, I'm going to talk about two topics in this presentation which are somehow related to each other, or I would say they are opposites of each other: attenuation and exaltation. Let us first understand what is attenuation. The definition is this: when the virulence of a pathogen is decreased, it is known as attenuation. So we already know what is virulence, right? Virulence is the disease-causing ability of the pathogen, isn't it? The degree of the disease-causing ability of the pathogen is called virulence. When this degree of disease-causing ability is decreased, when the disease-causing ability of the pathogen inside the human body is decreased artificially, it is known as attenuation. And this attenuation is mainly tried out right from earlier times for the human advantage, so that we can use these pathogens as vaccines. So here what we do is we decrease the disease causing ability of the pathogen and then inject it in human beings, so that when these pathogens enter inside the body, they will not cause the disease because their virulence has been decreased, but they can induce our immune system so that we become ready to fight against these pathogens. So such organisms or such pathogens which are alive but yet which cannot cause disease and stimulate our immune system, such preparations are called live attenuated vaccines. Now, how these attenuation is carried out? There are different ways of doing this attenuation. The most common way is growing the pathogens in an artificial culture media. See, we know that pathogens are best adapted to grow in the human body, and that is how they cause the disease, right? But when these pathogens are made to grow in artificial media, then not just once or twice, repeatedly if you grow them in artificial culture media, slowly they start becoming adapted to these artificial culture media. So once they get adapted to the artificial culture media, again you take that organism and when you inject it in human body, they will again have a problem in growing inside the human body. So this is how attenuation is done. Organisms which are adapted to grow in the artificial culture media will not grow that well inside the human body. So such organisms are called attenuated organisms. So let me just read this out. Growth upon artificial media results in attenuation of pathogenic organisms. In some cases, a very few passages in vitro will be enough to destroy the pathogenicity. That is, not just once or twice, you have to grow this organism in the artificial media for many times. Continuous subculturing must be done. Only then you can see such changes in the pathogen. So, such serial passages in artificial media has been tried with organisms like cholera, typhoid, tubercle bacillus, and also many other pathogens. So, we have vaccines now, live attenuated vaccines for cholera, typhoid, and tuberculosis, mainly by means of such serial passages in artificial culture media. So, one popular example for live attenuated vaccine is BCG. We already have seen the BCG under tuberculosis disease. Here I am going to concentrate only on how attenuation is carried out. So BCG is prepared from a strain of Mycobacterium bovis. So this live bovine tubercle bacillus Mycobacterium bovis has lost its ability to cause disease in humans. How? This strain has been grown in artificial culture media for many years by continuous subculturing, for many years, to be very specific, for 13 years, by subculturing it almost about 239 times, so that the virulent Mycobacterium bovis has become a virulent Mycobacterium bovis, and specifically it has been grown in glycerin potato medium. So in such an artificial medium, when this organism was made to grow for so many years by repeated subculturing, the organism has turned from virulence to avirulent. So such a strain is the BCG strain. Because the living bacilli get adjusted to make the best use of available nutrients in the artificial medium, they have now become less well adapted to human blood and can no longer induce disease when it is introduced into the human host. So whatever I said before, it has been shown in letters here. Since the organism has gotten adjusted to grow in an artificial medium, when you take that organism and inject it in human beings, they can no longer induce disease in the human beings because they have lost the virulence. But at the same time, they are similar enough to the wild type to provide some degree of immunity against human tuberculosis disease. Though they have lost their virulence, they are alive. They are still alive. So when you inject it, they might grow very slowly. And since it is growing there inside the human body, our immune system will recognize it as a foreign substance and will start mounting immune response against it. And that is how we get immunity against tuberculosis pathogen. So this is one classical example for attenuation. Now, apart from growing the pathogen in artificial media, there are other ways also by means of which you can attain attenuation of pathogens. I have mentioned three different types here. The first one is instead of using a susceptible host, if a relatively insusceptible or a resistant host is used, attenuation of the organism happens. See, here I have given one example of a smallpox virus. See, for smallpox virus, the susceptible host is human beings. So if it enters inside the human beings, it will cause the disease, smallpox disease. But if the same virus is injected into a resistant animal or an insusceptible animal, then the organism might become very weak in nature. So here the smallpox virus becomes attenuated by passing it in heifer. Heifer means it is the cow which has not given rise to cough yet. So if a cattle is used for injection of smallpox virus, then inside this heifer it might not go very well and it will become attenuated. So this is another way of attaining attenuation. The third way is, see, we know that temperature plays a very important role in the growth of any pathogen. And we also know that most of the pathogenic microorganisms prefer to grow at 37 degrees, which is the body temperature. So if you increase the temperature above this, for example, if you increase the temperature to 55 degrees or 50 degrees or 45 degrees, because beyond that, if you increase it to 60 degrees, it might just kill the organism. So don't go to 60 degrees, which is the lethal temperature. Instead, go to higher temperature, which might not kill the pathogen, but it might just attenuate the pathogen around 45 degrees or 50 or 55 degrees for a shorter period of time. If you expose the organisms, then the organism might become weaker after some period of time at this particular temperature. So this is another way of attaining attenuation. And the fourth way is you grow the organism in some kind of a disinfecting agent. See, no, we know that disinfectants like phenol will kill the organism in its presence, but at a particular concentration, right? So don't you grow the organism in a particular concentration of phenol so that it will just make the organism weaker in nature instead of killing it. See, we want the organism not in a killed form, we want it only in the weakened form, in the weak form, right? So you just grow the organism in increasing concentration of disinfectants like phenol so that the organism will lose some of its activities which is responsible for virulence. In fact, Pasteur used this formula, used this principle in producing the rabies vaccine. We know that rabies vaccine was first prepared by Pasteur, right? So what he did was he took the spinal cord of the diseased animal in where the rabies virus was there, and then he exposed it to caustic soda, which is again a disinfectant, and he just didn't use a concentrated caustic soda. He used different concentrations of caustic soda, and he tested the virulence of this rabies virus in different concentrations of caustic soda and obtained various degrees of attenuation of rabies virus under different concentrations of caustic soda. So this is also one way of getting an attenuated pathogen. The next concept is exaltation, which is just opposite to attenuation. See, we said attenuation is when the virulence of a pathogen is decreased, whereas exaltation is when the virulence of a pathogen is increased. And just like attenuation, this exaltation of different pathogens can be done in different ways. The first way is uh, you can enhance the virulence of the microorganisms by repeated serial passage through susceptible animals. See, I said when I told you about attenuation, the first way is repeated serial passage in an artificial media, isn't it, to decrease its virulence. Whereas to increase its virulence, you have to do the opposite. You have to do repeated serial passage through susceptible animal. Susceptible animal is in the animal where it can easily cause the disease. So when you do this slowly, day by day, its virulence will keep on getting increased. So this again, an excellent example for this exaltation was done by Pasteur again when he was working with rabies. He was doing a lot of experiments, how to decrease its virulence, how to increase its virulence, all these things he was doing. And what he observed was when he did repeat a transfer of this rabies virus in rabbits, he kept on injecting the virus in rabbits until the virus killed these animals within 6th or 7th day. Usually it takes longer period to kill the rabbits, but what he did was he kept on repeatedly injecting the rabbits continuously, and from one rabbit he used to take the virus, purified it, and then inject it in the next rabbit. So like this he kept on doing serial passages from one rabbit to another rabbit until this virus could kill the rabbits within a very short period of time, that is within 6th or 7th day. In fact, not just by 4 or 5 passages you can get this. He did about 270 such passages from one rabbit to another rabbit. Like this, 270 such passages he has done to enhance the virulence of the rabies virus. So this is one classical example of how to do exaltation. The next way is when you inject this pathogen along with another organism. By means of this microbial association, you can get the exaltation result. That is, you take a pathogen, along with that mix another organism which could, let us say another organism which can diminish our immune response, or which can inactivate our immune response. So if such a pathogen is injected along with our desired pathogen, the exaltation of our desired pathogen can happen. Okay. These are some other ways of doing exaltation. The third way is introducing the organism into the animal body along with another chemical. Instead of another microorganism, you can introduce it with another chemical like lactic acid. Instead of lactic acid, you can even use another organism which produces lactic acid. So this also might help the pathogen to increase its virulence inside the body and cause disease much efficiently. And sometimes you can increase the virulence by passage in vivo with the organism enclosed inside a collodion or a semi-permeable membrane. That means you are going to cover the pathogen inside a protective layer, and then you inject it inside the host, so that no no phagocytes or no other immune substances can touch this organism. This we usually do with vaccine. Sometimes we inject the vaccine along with an adjuvant, so that the vaccine organism will not be attacked by immune system very quickly, and at the same time the vaccine organism will be released in our body gradually and slowly, so that the immune response will be enhanced. But here what they have done is, you take a pathogen which can cause the disease. Sometimes what happens, when this pathogen just enters inside the body as such, our immune system, if it is really good, if it is really strong, it will not allow the pathogen to grow inside the body. So to make the pathogen get resistance from our immune system, we are going to protect it inside a protective layer. So this protective layer could be a semi-permeable membrane, or it could be a collodion inside which you can inject your pathogen and then introduce it inside the body. And sometimes what you can do is, you just inoculate this pathogen into an area in the body, like the sole of our feet, where you will not find phagocytes. Usually phagocytes will not enter such regions. So if you introduce the pathogen in these areas, the pathogen will not be encountered by the phagocytes, so that they can grow efficiently and can cause the disease. So you can select such areas for introducing the pathogen inside the body, where it will not be attacked by immune system very easily or very quickly. And the last way of exaltation is frequent consecutive transfer of the organism in a medium containing some kind of body fluids or blood serum, which will increase the pathogenic character of that organism. That means you grow the organism in a medium, and that medium should contain some kind of a body component, human body component like blood serum or some other fluid, so that the organism will get used to your body components, and naturally its virulence will be enhanced, and you can use it to cause the disease. So these are some way of doing exaltation.